Hello, in this video we will discuss about the development of chick. So embryological step we will discuss in this video. So incubation time is 21 days. So remember. So the gamete formation is the initial step we will discuss, and we will understand about the overview of the developmental stage. So here is the gamete formation, mean the sperms, haploid and egg formation, and these fertilize together to give a diploid zygote, and this is known as a fertilization process, which that is create the recombinant chick for evolutionary process and adaptation through environmental changes. So the fertilization to become a cleavage formation. In this way, the mitotically divide the zygote in this manner, and this is known as the discoidal cleavage in the chick due to the uh, due to the uh, uh, due to the zygote are present in the upper part of the uh, yolk. So the cleavage formation, after this cleavage formation, the blastomere formation will be occur, and germ layers will be formed, and in this way the gastrulation formation, and this gastrulation formation mean is known as gastrula, and this gastrula contain a epidermis, hypodermis and endodermis, mesodermis and endodermis, sorry. So the organogenesis will be occur after the gastrulation, and this is the organogenesis to become a growth and cell division will be occur. So the cell growth and cell division. So here is the egg. Let's begin to understand that here is inside is a zygote, and the shell is contain calcium carbonate, and this egg will be incubated, and after the fertilization and incubation we will discuss here. So after the incubation, so the normal temperature to become a more 36 to 38 degrees Celsius temperature required for the cleavage and the cell division. So in this way the cleavage formation, and this is the discoidal cleavage we will discuss here. So this is the a zygote, which that is the disc like structure you can see in this video, and in this way you can see that is the first cleavage will be horizontal or vertical on the basis of their angle, and after fertilization mitotic division will become a 2 cell stage, 4 cell stage, 8 cell stage, and more to become a morula formation. Morula formation is known as the packing and the group of the same cells which that create the fate for the development and create the new organ on the basis of the requirement on the region of the chick. So after the morula formation we will discuss here, you can see. So the morula formation is the mass of cell will be formed. In this way there is no inside is the any cavity, but later on the morula will become a blastoderm formation is the upper side, and yolk is here you can see, and this blastulation formation, will be occur, and this blastula form, and that's blastoderm formation, and this is the blastocele is the inner cavity which that is known as blastocele, and the segmentation cavity is known as the blastocele. So in this way here you can see this is the blastocele. This cavity will separate the blastoderm is the ectoderm and epiderm and hypoblast. So the here is the undetached cell is known as the zone of junction, which that will be undetached and attached with the yolk for nourishment. And in this way here is the gastrulation formation. In this way the germ layer will be formed. This germ layer is the here is a primitive streak which that create the next future spinal cord and brain through a Hansen's node we will discuss also. Here is a yolk, and here is the migrating cell is the red color showing this in this way, and imagination to create the primitive streak, and this is known as the gastrula. And in this way here is the migration outward will be occur. So the outward migration, and here is the epiblast. Epi mean the above, So this is the epiblast is become the mesoderm and ectoderm future, while the hypoblast is the future endoderm. Endoderm means which that is the digestive system and the other part of the body, while the mesoderm is used for the circulatory system as well as other digestive organ, while the ectoderm is the skin. So here is I am drawing again for understanding after 18 hours later. So you can see in this diagram to that is the hypoblast is here, and here is the primitive streak which that become a Hence's node which create the brain formation, and this is the invagination of the cell, and the migrating cell is the red color is showing in the figure, and this is the endodermal lining of yolk sac, endodermal lining of yolk sac, and this is in this manner here is you can see this is the endodermal lining of yolk sac is here, and this is the subgerminal space are present. This subgerminal space is important for separation of this discoidal part of the gastrula. So here I am drawing the frontal view, upper part of the view for understanding the Hence's node and the primitive streak which that is the future brain and spinal cord. And this is the primitive groove formation. This primitive groove is invagination of the cell and migration of the inside the cell is the migrating cell. And here is the primitive streak, and this is the primitive ridges. This primitive ridges is here, and here is the primitive ridges is looking like this is that, and caudal end and cephalic end. So the Hence's node is a cephalic end. So area of pellucida and area opaca formation will be occur. So here I am drawing the gastrulation, the neurulation formation. So this is the black color showing that the neural groove formation by the invagination after the 25 hours later. So in this way here is the notochord formation. This is the notochord, and this is the spinal cord formation, which that will be later on will become a neural groove will be bind in this manner. So here is the superficial ectoderm, and this is the heart rudiments are present in this way, and this is the notochord, and this is the neurulation formation, and this neurulation further you can see and understand the neural tube formation will be occur after the complete covering of the ectoderm, superficial ectoderm. So in this way here is you can see that's a forming heart tube, and this is the foregut formation here you can see in this diagram, and in this way the coelomic pericardial space formation will be occur here, and this is the coelom formation, and in this way these layers form extra embryonic membranes upper and lower. This is the extra embryonic membranes. So let's begin to understand after 5 years. Here is the allantois, amnion, and allantois, amnion, and chorion, which that is a different layer will be occur, and the allantois is the basically used for the nourishment, used for the excretion of the chick, while the later on it can be used. And on the other hand here is the albumen, and this is the yolk sac, and this is the basically is the after the 5 days to 10 days later on is the amnion is here, the white color, and allantois is the here you can see, and this is the albumen, and this is the yolk sac and the air sac you can see in this diagram. After the 20 days the chick complete develop, and the allantois will be decrease, and the amnion will be also, and in this way the allantois will be here, and this is the allantois will be decreased. So in this way this is the chick development.